What I'm going to talk to you today is actually an incomplete story, and I'm sort of hoping that through this talk some of you will challenge me and get me thinking in fresher directions than I have been doing so. So I'm a cellular neurobiologist who's also a geneticist, and if you look at neurons, that is the heart of how your entire brain works, in my opinion, the most important cell. And each neuron is a highly compartmentalized cell. So I'll give a little bit of introduction because not a lot of biologists here. A neuron is a cell which has as a receiving end and a sending end in its simplest form, where it receives information from either sensory system. So for instance, I can tell Rahul Siddharthan's wearing a check shirt, and that's because there's light hitting his shirt and then hitting my eyes, and my neurons are processing that information. And you have an axon which collects all of that information that comes from these various input areas. And right around here in the axon, early part of the axon, the cell body, you have integration of the information, and that electrical information goes to the synapse. Neurons in general can be very long-lived cells. So, if I'm giving a public talk, I'll always ask the question, how many of you have grandparents who are still alive? Mine is 101 and she's still alive, she's still breathing. That means a neuromuscular junction, that is the neuron telling the muscle to move, the diaphragm so that the lungs can take in and send out air, are still functioning, and that's a 100-year-old cell. So these are very, very stable structures, and part of the reason why neurons can execute all of these complex functions and have this kind of stability comes from its underlying architecture, and that comes from both the cytoskeleton, and it's able to function because of subcellular processes that occur. So in a neuron, you have tracks or molecular roads called microtubules on which cargo move and reach the synapse, and these cargo are called synaptic vesicles, but they can also be mitochondria. And if anybody has studied any school-related biology, even up to 10th grade, you will always think of mitochondria as powerhouse of the cell. It does a lot of things besides that, but it's also the powerhouse of the cell and generates energy. And this kind of movement, either from the cell body to the synapse or from the synapse back to the cell body, is taken care of by molecular motors which use energy to move cargo. And the reason you need to move cargo is because majority of the cargo is actually made here. And neurons, as I said, can be very long cells. For instance, you can be a giraffe, or us. We have a 6 feet foot person will have a neuron that starts in the spinal cord and goes all the way to the big toe. And big toes, I've learned to recognize after seeing cricketers and footballers, are actually very important if you want to do something meaningful in that sport. Okay, so you need this kind of transport process to keep it going. The transport occurs through underlying cytoskeletal architecture, which is made of these molecular roads. But in addition, there are other cytoskeletal architectures whose function is not very well known, and that's, for instance, here shown as actin, which is present in these ring-like structures, which was described not more than 7-8 years ago. And the idea is that they provide potentially some kind of support to the neuron to keep the plasma membrane or the cell wall up, not cell wall, the cell membrane up. So in this complex cell, and the many things that move from the cell body to the synapse and the synapse back to the cell body, one of the cargoes we look at is the powerhouse of the cell, which is called mitochondria. And mitochondria are these organelles, which I won't tell you a whole lot about. They have an outer membrane, they have an inner membrane. These have complex proteins on their inner cell membrane which generate ATP. They also generate reactive oxygen species, which are very, very detrimental unless they're taken care of, and they're partly taken care of by other cellular mechanisms and by the mitochondria themselves, but they need to be taken care of. So you have this network-like architecture of mitochondria, which is all attached to each other, and they're very supposed to be quite dynamic in the cell body. But when you look in the axon, what you see is a molecular motor will attach to a mitochondria and it will move it. But you'll also see in every neuron that anybody has ever looked at, majority of the mitochondria actually don't move at all. something like 80% of the mitochondria in a given time when you look, 80% of them don't move at all. You now know that they are anchored to the cytoskeleton. They can also be anchored to actin. They can be also, so they sort of possibly these anchoring agents are what keep them in place. And of course, they do all the normal transport-dependent phenomena. That is, they move along microtubule tags, hop onto another microtubule tag, get anchored, come up to the synapse, where they also provide energy. And their presence in the synapse is actually pretty important for function, because you need a lot of energy at the synapse, and you also need, just a minute, you also need some sort of calcium buffering, and which is another very important ion that mitochondria deal with. Yes, Ronnie? That's certainly one of the ideas that are out there, and there's some proteins that are known to do that tethering, syntaphyllin and So in other work which we have done with Gautam Menon, we actually think cargo crowding is a very important factor in regulating axonal transport. And what we see in our own movies that every time there is a mitochondria present, cargo are jammed on. Synaptic vesicles, membrane cargo are jammed on both sides of it. But you have to recognize that eventually they will resolve. So it's not a permanent traffic jam. But yes, I think those are really physically crowded regions which can lead to more cargo halting. And in fact, in Gautam's simulations, the way he models the mitochondria is as a block in transport across multiple microtubules, which makes sense because it's a large organelle. Okay. So, most people who look at mitochondria who are biologists will ask questions about its function, and I have no idea why this occurred to me, but what I wanted to understand was what controls mitochondrial numbers in neurons. And I think it came from actually when I first made this transgenic line as a postdoc, I was very intrigued by it. I made it because I wanted it as a contrasting cargo, as a control cargo for synaptic vesicles, which I was looking at, and I was very struck by the arrangement in neurons. And I'll just show you, these are not the best images, but they're still very nice images, and I always show them because the first graduate student who worked on mitochondria, Guru was the one who took these pictures, and he unfortunately died in a road accident before he could get his PhD, and this is my way of always reminding myself and honoring his work. Okay, so what I saw was there were occasionally mitochondria which were very close to each other in the neuronal process. There were of course some, as you wouldn't expect, in the synaptic regions of the neuron, which had been known, and you know that mitochondria function in the neuron. But what I was very curious about was to look at the distribution in the neuronal process. And two things struck me when I looked at it, is that no two mitochondria are very close together, and none of them were very far apart. So that was the starting point about where sort of motivated me to look at it. And we decided to sort of start tracking mitochondria along the entire neuronal process. Okay. And what Anjali found, she was a teacher who was also doing a PhD on the side, what she found was that as the animal grows, the number of mitochondria increase, such that essentially the density of mitochondria as the axon is increasing remains the same. So this site of somehow there is some sort of number counting that's happening in the neuron. And so the first problem that we took on was to actually ask a question, where are mitochondria added in the neurons first, and the second, are the positions along the neuron regulated? And of course, as a biologist, I absolutely no clue how to go about it, so it probably took us longer to get here than it should have. But the first thing, one of the first things that we did, not one of the first things, after a long time we actually did this, we measured the inter-mitochondrial intervals that are present in the neuron, in any given neuron, and plotted them all. And what we saw was this was sort of the distribution, and these are the posterior touch neuron cells, and these are the anterior touch neuron cells. It doesn't matter what those neurons are. There are no synapses in these regions, and the axon had this particular distribution. And Sudeep, who was a postdoc in the lab and was a physics PhD student, who had done a PhD with Venkat in physics in IISc, came and said that one way for us, I said, oh, I'm sure this means that it's regulated, but how do you actually show that? So what he did was actually a simulation where he took a neuron of X length, which we knew what that length was, used the density that we knew to be constant around 5 and just did a very simple simulation and said, in the simulation, what are the inter-mitochondrial intervals? And we saw that that curve was an exponential fall. And we've gone on to do additional work with Sandeep, where he actually did a mathematical calculation of how it should be, and that's the superimposed red line with my student currently, Shweta. But what this data led us to believe, led us to recognize, is the fact that you didn't have too many mitochondria too close together was what that regulation was. That is, no two mitochondria could come very close together, despite there, of course, being this long tail. Okay. If that was the case, very simply we ask the question, how do you determine where the mitochondria are added as the axon grows? Because the density remains the same, but the number of mitochondria, of course, increasing, scaling with the length of the neuron. Or, how does short-term saltatory mitochondrial transport lead to long-term regulated distributions? And for this, yes?
You treat the axon as multidimensional so that at any position there's only one mitochondria
yes.
stacked like that?
Yes. Yes, and that's what we see. Actually, if you do, so I looked at, so when I was in Mike's labs doing my postdoc, I had access to the EM images of John Mike, where he reconstructs the entire nervous system, and I looked at every single section that I could get. I also did my own EMs, and I see that every location in the axon will only see one mitochondria. I actually never saw a case where there were two mitochondria right next to each other, but when you look at live imaging, you see one mitochondria cross another one, so you will occasionally get that, but I didn't see it. So I think in each location there's one mitochondria. Same thing was seen by Miriam Goodman, same sets of neurons when her postdoc Jose looked at it. So I think I feel fairly confident in thinking of each location, each bright spot is one mitochondria, or largely one mitochondria. Okay, so what Sudeep did was we both decided the best way to address this question is to develop a microfluidic device, image the animal over time, image the same neuron over time, the same animal and the same neuron over some 36 hours, and we feed the animal while that's going on and then watch to see where mitochondria are added. Okay, so this was a PDMS device that he designed. And essentially what you do, if I can, you have one layer and a second layer that you have on top. That went by very fast. Let's see if I can. What you end up trying to do is to put an animal in through the input, which will then come down into the imaging layer, and then it can move freely. And through those same pipette tips where you put the animal in, you also put in food supply so that you can actually feed the animal. And then you have these additional trapping devices where you can therefore trap the animal in just a little bit of this restricted space instead of having it freely move about. And then when you want to image it, you trap the animal, look at that image, wait a few hours, look at it again. So we did all kinds of imaging using this device. We used to sort of image every, continuously the way we would image for 20 minutes, we used to image every hour, we used to image every 3 hours, and we built up a long-term data set where we could look at animals as they grew and the same neuron on the same side of the animal as they grew. So here would be a free worm, here would be a trapped worm, and you would capture these images. Getting that kind of data, we could now tell, we for instance knew that this particular neuron had a branch over here and that branch point always had a mitochondria. So we had the end of the neuron as one tip, one fiduciary tip. Another was the beginning of the neuron that we knew was invariant. And then you can track over time and see where mitochondria are added. We say a mitochondria is added when we see it present, absent in the previous time point, present in the next time point, and continues to be present for 6 hours after that. So then we know that we've actually added a mitochondrial site as opposed to a mitochondria which just moves around temporarily. Doing that kind of data, what we saw was at 0th hour, this was our distribution of inter-mitochondrial intervals. At 24 hours after imaging, this was the distribution that we saw with inter-mitochondrial intervals. And what this actually showed us was that, or suggested to us, that you were not having addition take place in one area of the neuron, right? So if you saw that all the additions was in one end or the other, then this particular small bin size should actually be much, much higher, whereas it actually came down, the number of inter-mitochondrial intervals. In addition, we looked at where the additions took place, and what we saw was two adjacent mitochondria had to go apart at least by 20 microns for a new one to be added. Of course, it had a tail again, but very rarely would you have an addition of a new site if you were less than 20 microns apart. Addition can take place, as we thought from the distribution itself, anywhere along the neuron. It can be in the early part of the neuron, it can be the middle or the tip, and the deciding factor appears to be how far apart are two mitochondria. This immediately gave us the idea that there might be some code which one mitochondria sort of sends out to another mitochondria. And this conversation, we don't know the answer to this question, right? This is still very much in the design phases of it. But there must be something which is a signal between two mitochondria, either a repulsive signal which prevents two mitochondria from being right next to each other for long periods of time. And of course, there must be some sort of anchoring factor, and we of course turned to the vertebrate literature where there had been reported anchoring factors, but none of those anchoring factors were encoded in the C. elegans genome. Okay, so let's move a back step, which we get asked frequently, is that you could just be populating these new sites because of existing mitochondria break apart and they just stop next to each other. So we went ahead and tried to measure what we could see with respect to that kind of data. When we bleach an existing mitochondria, so we can label it with GFP and we bleach it, we can look at mitochondria which come and stop and fill the entire volume. We don't know if that's fusion. We can also see, and I think this will repeat, bleach it, one mitochondria comes past, gives some and some more leaves behind. So we can certainly see events where one mitochondria breaks apart, but we never seem to see an event where one mitochondria breaks apart and the remaining portion just gets stuck somewhere close by. It continues to move. So yes, mitochondrial turnover does take place, but majority of it is crossing an existing mitochondria without doing anything. There are a small minority of events where you see in the anterograde direction when it's coming from the cell body where you pause and split. So we think there is potential for the new mitochondria to come from turnover events, but it's equally likely that new mitochondria come from being fed from the cell body. I'll skip this and say, we then ask the question, how are these distributions regulated? And it's been a long journey, and I'm not saying we have any answers, but I'll tell you how we thought about the question. Very simplistically, we thought that some of the probable regulators, motors might be very, very important, because motors have to move mitochondria, and then motors will halt mitochondria at various places because the motors can get inactivated. What we actually saw, and here was our previous slide, was looking at various motor mutants. You could change the number of mitochondria in the axon, you could change the transport flux, but you actually did not change the distribution very much. So in that, no two mitochondria are very close together, and I won't show you that data here today. Our second thought was the cytoskeleton. We spent an enormous amount of time looking at the microtubules and got nowhere. Neuronal function did not seem to have an effect. ATP, we are not still sure that we've done the right experiment, and calcium seemed to have a modest effect. And we have not tested the multiplicity of metabolites that the mitochondria produce, like succinate and ROS and so on and so forth. They're all things that we should be looking at. But when we decide to look at the cytoskeleton, the key thing is to look not just at the microtubule, which we, as I said, spent an enormous amount of time looking at and we didn't get anywhere with it, but to also look at actin. Let me just show you a movie of how actin looks in a neuron. So you label a protein that can bind to actin, and then you image the animal or image the neuron inside the animal, and what you will actually see is actin showing all these blips, and movements and places which are stationary and which don't move. You can take these movies and convert them into kymographs, and what you will see is that you have these actin-rich regions that you are seeing, places which are less actin-rich, and you have these trails which actually actin is polymerizing or shrinking. What Parul Su, the student who actually worked with me and also with Gautam when we were trying to look at cargo crowding, she saw that actin was actually, mitochondria was actually present at actin-rich regions, or majority of the mitochondria were present at actin-rich regions. Here's quantitative data done by Shouvik Modi in my group, where he showed that majority of the mitochondria were present in extremely long-lived stable architectures. Some were present at more shorter-lived architectures, and sometimes you'll occasionally get a trail coming out from where a mitochondria was docked. So we knew that mitochondria is present at actin-rich regions. We also knew from Shouvik's work that mitochondria paused when it was moving at actin-rich regions, right? So actin was a place where majority of the pausing takes place, and very little pausing took place outside of the actin-rich regions. So this is great. So we thought that actin could be one of the factors which actually result in trapping mitochondria at specific locations. The only confounding factor for us was that actin is present everywhere, and this easily tells you this on your previous chymograph that you looked at. Right here, you have actin present absolutely everywhere. Actin here is in red and the mitochondria is in green. So you don't have any specificity. You have only about 20 mitochondria in a 400 micron axon, but actin, and here's your scale bar, is just everywhere. So that can't be the only answer. However, what we also saw, and we still think actin plays a role, is when we started looking at experimental data across different developmental stages. And what we saw was that the number of mitochondria which were very close to each other was lowest in adult animals and probably highest in young animals. And you can look at the Fano factor in a 20 micron base size and you see the same signature. In L1, the Fano factor is significantly higher compared to your other developmental stages, and your simulation, which is tending towards random, will be close to 1. And what we also saw was that actin changed and the dynamics changed as the animal grew. In very, very young animals, you had very long-lived actin, which reduced as the animal grew and sort of settled down, and least amount of long-lived clustered actin was in adult animals, which was surprising to us if you're thinking of a model where stable actin will trap mitochondria. Here's another way of looking at the same data slightly differently, which has some location specificity. What you see is that actin-rich regions is present very highly in certain regions of the neuron in very young animals, but as you get older, this actin-rich region is distributed everywhere. This suggests that actin itself redistributes much like the mitochondrial redistribution that we see occur. Are these two related? So to do that, Shouvik developed this reagent where he could dissolve actin inside animals in neurons. And what he saw doing that dissolution, so this is usually your actin-rich regions. What you see is now those actin-rich regions are gone. You can take a movie, you'll not see a single trail, you don't see any actin-rich regions. That the total number of mitochondria did not change, which was a blessing, because that told us that motor-dependent transport and putting mitochondria in the axon is not affected. But what we saw was really the Fano factor was changing, and the number of mitochondria, which were very closely spaced, were much closer. But one thing which struck us, which suggested that actin may indeed play a role in trapping things, however, it's probably only one amongst a multi-component system. And the reason it's only one among a multiple component system is because you still do not get a large number of mitochondria present very close together. The inter-mitochondrial intervals, if you do a simulation, is an exponential decay, which a largest number of mitochondria are placed very close to each other. So here's where we came with the story. We're sort of looking at calcium and other things at this point of time, but for the rest of my talk, for about 10 minutes, I'll take you into a purely biology arena, which was an utter surprise and something that we did not expect. So we were looking at all of these actin experiments. I said, you know, Shobik, we have to do a control where we don't have any mitochondria in the axon, and just we need to make sure that actin looks okay, because, you know, actin is present along with mitochondria. Formally, there's a possibility that mitochondria will affect actin. Although in the literature, you have a lot of examples of actin affecting mitochondria, except in the developmental context, where you can see that mitochondria through ROS can do something to the actin structures. I said we are not looking at a development context, we are looking at adult animals, but nonetheless this is something we should do. So we took advantage of a mutation called RIC7. It doesn't look like anything because it's present only in nematodes, and what you see in wild type animals, you will have mitochondria along your neuronal process, and you have these very nice beautiful actin dynamics, stable structures and trails. When you don't have a single mitochondria, and I mean single, you don't have a single mitochondrial neuronal process, all the trails vanish. Now we do some cool genetics and we drive mitochondria in the axon by directly attaching the motor to mitochondria through a mitochondrial protein. You drive mitochondria back into the axon, you restore actin dynamics. So this essentially told you that actin dynamics in an axon, which was a complete surprise for us, was controlled by mitochondria. So here's your lifetime of all the various actin in wild type. What you see in RIC7 is everything becomes extremely hyperstable, and now when you reintroduce ectopically mitochondria, you now restore it very similar to wild type distributions. And now the simpler way of representing the data, you have wild type, you have trail density. In RIC7 you have hardly any trails. You put the mitochondria artificially back in this mutant background, you can get actin dynamics again. So there were two questions when I started, when I wanted to look at, you know, number counting in the axon and say how do you control these numbers and how do you position them in the axon. But any good biologist is going to ask, does it have any meaning? So when I saw this, I said, and we had been trying this for a while in different contexts, I said this gives us the perfect example to look at the role of axonal mitochondria in behavior. Everybody's looked at synaptic mitochondria, we must look at axonal mitochondria, and here's an attempt to do that. So the way we do this experiment is, and I think anyway, I'll tell you the data and show you the assay later. The way we do the experiment is to actually touch the animal and see whether it responds or not. And this touch sensory behavior depends on these neurons that we have been studying. So in wild type animals, each time you touch the animal, it's an escape response, it moves. In RIC7, which has no mitochondria in the axon, including at synapses, your touch response is reduced. When you artificially introduce the mitochondria, you can now rescue that function. So we can say mitochondria are necessary for touch responsiveness, but we have no way of knowing if it is actin which is important for touch responsiveness. So we asked this question because there's been a lot of work and general thoughts about how actin-rich regions which are stable are possibly important for neurons, that they may actually hold up the membrane, they are associated with other proteins and give structural stability to the axon, and nobody to this date knows what actin trails do. And Ala was the first to describe that there were actin trails in vivo in animals and not just in cultured neurons. And since in our mutants we could actually see that actin trails were affected, this gave us an opportunity to ask to distinguish between the role of actin and the role of mitochondria. And to do that, I've simplified a couple of years of work in one slide, and I won't tell you much about this except to say the following. When we looked at a mutation which produced mitochondrial ROS, that by itself gave us a lot of trails. When we built a double mutant of that, that is, you took out the ability, you made now a lot of ROS, and you also had no mitochondria in the axon, your actin trails were not reduced. But when you looked at, when you didn't have mitochondria and restored mitochondria back, you put the trails back. But when you removed SOD2, that is, you affected the ability to produce ROS, in fact you made much more, this completely was abrogated. So in these animals, you continue to have mitochondria in the axon, but you don't have trails. So you go back to the gentle touch assay, where you touch the animal and it runs away, it's an escape response. Or the way that actually the experiment is done is you touch the animal in the posterior and anterior and count how many times it responds. And in fact, the last test, it did not respond very well. And what you see is the following. Sawtooth animals by themselves can respond to touch. When you look at RIG7 mutants or sawtooth RIG7 mutants, or you remove sawtooth in animals which have mitochondria, you abrogate touch responsiveness. So now we have decoupled, we don't have an answer what mitochondria does, but we can at least say mitochondria-driven actin dynamics are very important for touch responsiveness. So axonal mitochondria may not themselves be doing anything, even though we know that they take up calcium. We've seen that in the lab. We know the mechanism by which calcium is taken up. But we can say that at least this behavioral response, this sensory behavioral response, depends on mitochondria-driven actin dynamics. So the next question then to ask, is dynamic actin sufficient for neuronal function? Do you actually need the presence of the mitochondria at all, even though the previous experiments suggested that it was mitochondrial-driven actin dynamics? So what we did over here is again a very, very simple experiment. We took a particular genetic mutation, which is an activated form of Rho, which we know controls actin, and we just expressed it in these such touch receptor neurons. And what we saw with trails, and if you just look at this part of the image, that in RIG7 you hardly have any trails. This has no mitochondria in the axon, and when you express this activated Rho, your trail number increases. By itself, it doesn't have a huge effect on wild type. And now when we look at the behavior, what we see is the following. RIG7, you have abrogated behavior. When you restore actin trails, you improve behavior. And just having that particular genetic mutation in a wild type background does not seem to affect behavior. So our takeaway here is the following. We find that mitochondrial distributions in axons are regulated. Addition of mitochondria depends on a threshold of separation. It has to be at least 20 microns apart before a new one comes up in that location and be a long term. We know that mitochondria are present and pause well near moving at actin-rich regions. We think that actin is one part of the component which controls inter-mitochondrial intervals. And dynamic axonal actin, which is a complete surprise for us, depends on mitochondria and is necessary for behavior. We are now doing work, or we are going to begin doing work, where we will now ask, like good geneticists, questions of, we've now removed actin, and now can we remove these other metabolites and find that this inter-mitochondrial interval is affected. But what it seems very clear so far, we have small effects with calcium, we don't know yet with ROS and ATP. We've done what we think are the right experiments, but it requires more verification. We think that the spacing between mitochondria is because mitochondria that are moving pause at actin-rich regions. There is some sort of encoding that happens over there where the mitochondria sort of wait to see whether they're in a trough of some sort of signal between two adjacent mitochondria. If they are in a trough, I think this particular interaction is stabilized, and if they are not in a trough, I think it can continue to move. At least that's what we think so far. So the neuronal cell biology just became a lot more complex, where we now started to need to think about dynamic actin and why mitochondria, while present at actin-rich regions, also controls dynamic actin. And from the behavioral angle, we now know through a collaboration that we're doing with a group in China that these effects are through electrical synapses and not through chemical synapses. So most of this last part of the work was done by this extremely talented postdoc Shovik, who is now head of research in Asia Labs. and a very talented graduate student, Sneha. And these are all the past members. Guru, who started the project when we knew nothing and was very brave. Sudeep, who did the initial simulations and developed the microfluidic devices. Anjali, who did this painful job of imaging and analyzing lots of mutations and looking at all the developmental stages. Jyoti, who got some of the turnover data with mitochondria. Anushila, a master's student, who did some of the motor mutations and their role, and Nikhil as well worked on the same project. So here are our collaborators. Some of our most recent work when we're looking to understand the distribution is done with Sandeep Chaubey, and these are other collaborators. And with Gautam Varuni, actually tried to set up a pipeline for doing automated analysis, which did not go anywhere. Unfortunately, it would have been really nice if it had, but we spent a decade trying to understand this problem and made some tiny headway. So thank you for your attention.
So if you have any questions, please, yeah. One minute. If you have any questions, please, yeah. Pinaki.
In the experimental results for the inter-mitochondrial distance, I think the distribution sort of showed a increasing and decreasing trend.
What do you mean by increasing and decreasing trend?
So, I mean, it was not like a clear exponential decay as shown in the simulation.
It's not. That's the whole point. It's not an exponential decay. What it is right here. Let me try to find that data. That there are very few mitochondria which are present very close together. Right? This is your experimental data. The very few mitochondria which are present close together, this portion may still be an exponential decay, and we are doing some simulations now to see if we can shift it. If you take these out, does it only happen? Is the repulsive effect acting only in the first, 10 micron apart bin, and after that it falls apart? It's possible. It's very, very different from what we see with our simulations, where majority of the mitochondria are actually present close together. And that's where the regulation occurs. And if you look at the Fano factor calculation, when you remove actin-rich regions, you lose some of that. In fact, more mitochondria now start coming together, closer together. So we think that actin is one part of the component where you have to maybe your position is stabilized. It's not as if your mitochondria are unable to read the signal, but they are stabilized when you have actin-rich regions, and it holds them in place. At least that's what I'm thinking about. I could be completely wrong, but it's a reasonable model and it's testable.
Any more questions? Okay.
So this distribution function is very interesting, because if you have very disordered systems, like a random matrix kind of thing, then they have this peak-like structure.
Yes.
And the other one is reminiscent of stochastic, very far apart, the dilute kind of system.
I don't know if it would be dilute. I mean, that's why I said the density is regulated. You can't seem to go below a certain point, right? So what I can tell you is that anywhere between 4.5 to 6 is what the wild type density will be. If you look at behavior, you need at least 3.5 micron per 100 microns. Okay, and it's a 400 micron length axon for you to be able to restore behavior. If you fall below that, you don't do very well. And there are other reasons which I won't get into, the biology reasons, but I think we are able to uncover the role of ROS, etc., when we have a sensitized system where we have a density of 3.5. So I don't know if this is stochastic. All I can tell you is there is some sort of regulation which prevents two mitochondria from being very close together. Actin is part of the system, but something coming from mitochondria is also part of the system. We don't know what that is. We suspected it would be calcium, but we have to do the right experiment to show that it's calcium. But it could very well be ROS, it could very well be ATP. There's one group which is published and says it's ATP. I look at the data and I'm not convinced. Our own data don't support that.
Yeah, Santan's question was sort of mine also. So, if I understand right, the fact that the belief that they get held into actin regions explains why they don't come together in actin-rich regions, but they also don't come together in actin-poor regions, and therefore there is some other mechanism for that.
Yes.
And
that's what I deduced. Actually,
Right.
the deduction that there are some other mechanisms comes from our long-term imaging data, which was where you see that a new site does not come unless two adjacent mitochondria at least 20 microns apart. So I think that's the strongest piece of data.
So my question, maybe more for Sandeep, is in the simulation. If you simulate particles with some short-range repulsive force, or
Has to be done.
and maybe two different repulsive forces, one for the actin regions and one for the non-actin
The actin
regions.
region is not a repulsive force,
It
it's
is
an
just a-
anchoring force,
Yeah,
right?
but
So
no-
it's a holding force.
Okay.
Yes, that's the hope that
Right.
Sandeep can
Okay,
help
maybe
with
not
that.
a force, but just a constraint.
Yes,
That,
a constraint. I agree. Yeah, yeah, yeah.
Yes. But just to see if your graph looks more like the experimental graph, if you put that in.
Yeah, that's what he's been telling me to do. Even, yeah,
Absolutely.
we are baby steps.
So, oh,
I was just wondering, how does the synaptic activity affect the migration of mitochondria?
So we haven't looked, have we done that? Yeah, we did one experiment where you don't have vesicle fusion taking place. It's a hypomorph, so it's not all gone. And we didn't see a big effect on mitochondrial distribution. Now, many of these things will have an effect on mitochondrial movement, which is why I said upfront that what I was interested in is the steady state distributions that emerge, right? I know it's an unusual question for a biologist to be interested in, but I think it's important that we look at that, because I think we have seen and we will continue to see small changes in axonal transport of mitochondria if you play with the synapse, if you change some other thing. But what's really remarkable is how refractory the overall general distribution snapshot taken at any given time is despite these changes. That tells you you have a very robust phenomenon in place. And by no means is this only for this neuron. So when people have looked at neurons in culture, or even zebrafish neurons, and they've done different things, where they will try to they will express some disease molecule over there. And their goal is only to look at, what is changing between the disease situation example and the mutant. But in each case, you see that the density of mitochondria is actually regulated. So for any given neuron type, I think this is going to be a very common and fundamental mechanism, if we find it out.
yeah.
Yes, so we've looked at that. So we looked at DRP mutants. So we basically looked at fission and fusion mutants. In fission and fusion mutants, you can change the individual size of mitochondria along the axonal process, but the distribution remains the same, which really tells you how robust this is. You can say, I don't think people can argue with this data. I think there is some regulation preventing too many mitochondria from being very close together if our simulation is correct, which I think it is now. Oh, you mean it's because it can't move? So it's a very nice idea, but what we have seen, and you must have seen in our movies as well, there are two types of mitochondria that we see moving. One which is much more tubular. So let me see if I can find that movie. Okay, some which are much more, circular, like every other mitochondria, and they'll do this, but they'll also often move across. And then we have something which is much more tubular, which is what you see here, which tend to break apart much more. That's the only distinguishing feature we have found so far. Rounded, nice, what look like bright fluorescence. Of course, we don't know what the size is, but we can at least say that it is not below a certain size. Seem to move perfectly well. So my thing is they have either a lot of motors or they're like a bully pulpit, they can push things out of the way. I don't know what it is, but they are able to move.
So if there are no more questions, we'll thank the speaker for this beautiful talk.
Thank you for your patience.